Here is the second part of the slideshow about cellular respiration, our chapter 7. So we saw previously the glycolysis and the citric acid cycle. So now we will see the electron transport chain, or how you can make ATP from high energy electrons, and basically the electron carriers that will move these electrons towards the electron transport chain. The electron transport chain is a name for a group of proteins that are located in the inner mitochondrial membrane and that will be able to carry and move electrons as well as protons. So a simultaneous movement of electrons and protons will happen in the electron transport chain, and the high energy electrons will progressively release their energy, and this energy will be converted in order to pump protons against their chemical gradient, so that they will end up and they will build up in the intermembrane space. Once they build up in the intermembrane space, they will be able to form a big gradient that will be used by another protein to form ATP. Basically, they will use the energy of the strong proton gradient to synthesize ATP from ADP and PI. So basically use the energy, the kinetic energy, in order to convert it into a chemical energy. So form a high energy bond between ADP and PI that will end up in ATP. So this coupling of the proton translocation from one side to the membrane of the from one side of the membrane to the other side of the membrane, and the use of this energy to synthesize ATP is called chemiosmosis. So for each pair of electrons that will be given by an ADH, you will be able to form 3 molecules of ATP. When the pairs of electrons are given by FADH2, it is only 2 molecules of ATP that can be done. But that's still quite a lot of energy that can be done by protons moved through the ATP synthase, which is the enzyme that will be responsible for the synthesis of ATP using the energy from the proton translocation from one side of the membrane to the other side. If we look at this in picture, you see here the 4 main components of the electron transport chain. And let's first have a look at the first complex, the complex 1. It is a complex of proteins that will be able to extract the H from NADH. Basically, the H will be separated into 1 proton and 1 electron. So a H plus and an electron make together a H, but when they are split, it can make a proton and an electron that are separated. And what will happen is that this proton and electron will cooperate. Basically, the electron will release its high energy in order to move and the energy will be used by the complex 1 to move the proton down into the intermembrane space, which is where there is a high amount of protons already. So because it is against its concentration gradient, it needs energy, and the energy is provided by the electron that has a high level of energy, especially here at the beginning of the transport chain. Once this is done, the electron will be taken in charge by the coenzyme Q, okay, or CoQ here. So it will be carried, and in fact the CoQ will be a kind of shuttle that will carry the electron directly to the complex 3. So it will avoid the complex 2 and go directly to the complex 3. When the electrons come from FADH2, in contrast, they arrive directly in the complex 2. So when protons are coming from NADH, they come in complex 1. When they come from FADH2, they will come in complex 2. Now, they end up in the same place. They will end up in complex 3 once the CoQ has taken them and shuttled them to the complex 3. Once in the complex 3, what will happen is that again the same thing will happen. The electron that still has quite a lot of energy will be able to release this energy. The energy will be used by the complex 3 to pump the proton against its concentration gradient into the intermembrane space. So you see that the intermembrane space starts to build up with protons. Again, the electron is not done. It will be taken in charge by another molecule, and this time it is the cytochrome C. It will be taken in charge by cytochrome C that will shuttle the electron to the complex 4 here, and once again, the leftover energy that is still contained in this electron will be used to move another proton, and finally, once the electron is almost totally out of energy, it will be accepted by a final acceptor, which is oxygen, that will also take another proton, in fact 2 protons, and it will form a molecule of water. So the formation of water is the final product of the electron transport chain on the side of the matrix of the mitochondrion. We also say that oxygen is the final electron acceptor of the electron transport chain. Now, we have, with this process, build up a lot of protons in the intermembrane space. So there is now a very strong gradient of protons that is with a high amount of protons in the intermembrane space and very few protons in the matrix of the mitochondrion, because we constantly take them to bring them in the intermembrane space. So this very strong gradient can be used, in fact the kinetic energy can be used in order to produce another form of energy. So this is exactly what will happen in the ADP synthase, that will take the gradient, in fact that will move the protons in the direction of their concentration gradient, so down their concentration gradient, but while providing an opening to the protons and allowing them to move on the side they want to move, it will also activate the synthesis of ATP. It will basically use the kinetic energy to form the bond between ADP and an inorganic phosphate, so that it will end up in the ATP molecule that carries a lot of energy. So this is, in fact, the use of the gradient of protons to form ATP is called the proton motive force. So it all says what it has to say. It means that you use the motive force, in fact the force of motion of the protons to make ATP. So basically the concentration gradient or the chemical gradient between the matrix and the intermembrane space will create a gradient of pH. It will also, because you remember that protons are acidic, it will also make a gradient of charges, because protons are positively charged, so it will create an accumulation of plus charges in the intermembrane space, so a difference of charge, and this can add up to the gradient of pH and together create a real force, and this is what is called the proton motive force. It is abbreviated in delta P to be calculated by experts. We won't do that in the course. Now, just a closer look at the ATP synthase, which is a pretty amazing molecule. In fact, it's a complex of proteins, and as you see here, it has in fact 2 main subunits. One subunit that is called the F0. The F0 subunit is a channel that will allow protons to move through. However, while moving through it, the protons will rotate the F0 subunit and it will transmit, in fact, the rotational energy will be transmitted to the other subunit, which is called the F1 subunit, that will use the rotational energy to catalyze the synthesis of ATP. So the 2 subunits will work together in order to use the the physical motion energy of rotation to make the molecule of ATP. So if we summarize the flow of energy in cellular respiration from a glucose molecule crossing the membrane of the cell, we see that the glucose will be able to enter in the glycolytic process and form two molecules of ATP. Then it will move to the mitochondrion where the citric acid cycle will be responsible for the production of two ATP molecules. And finally, the electron transport chain will give rise to the remaining 28 ATP molecules for a total of 32 ATP, which is a pretty good yield for a single molecule of glucose. This is if the glucose molecule is totally oxidized, which means totally broken down into CO2 and H2O molecules. So here a summary again of the yield by oxidative phosphorylation compared to the substrate level phosphorylation that we had previously. You see that it is the most part of the production of energy from the glucose molecule. What happens now if oxygen is not present? If oxygen is not present, then you cannot have the final acceptor in the electron transport chain, and as a result, you cannot produce energy from oxidative phosphorylation. So now it means that you will have less energy produced from the glucose molecule. However, one problem will arise when you have only the glycolytic pathway. It is that yes, you will produce ATP, so you have what you need from the glucose. However, you don't produce a lot of ATP, just two molecules, and you will have an accumulation of pyruvate molecules that will accumulate and build up in the cell if you don't get rid of them, plus the formation of the NADH or the electron carriers will also take a lot of importance here, and what will be problematic is that you may end up being short of NAD plus if you only rely on glycolysis. So for example, imagine that you're doing a very strenuous exercise, you have few availability of oxygen, then the limiting factor will be the presence of NAD plus that is needed to go on with the glycolytic pathway. So what happens in this case is that the pyruvate needs to be removed. You need to make more NAD plus. So what will happen is that H plus will be removed from the NADH, and together they will form acid lactic with the pyruvate, so that you can replenish the store of NAD plus, and at the same time get rid of the pyruvate. This produces lactic acid, so this is the end product if you have glycolysis happening in absence of oxygen, so no mean to get rid of the pyruvate by the mitochondria. This is what you will see in animals and bacteria, and it is a very efficient mean to remove pyruvate and replenish NAD+, so that you can form ATP very quickly. It is a very quick and very efficient mean to produce ATP. However, you don't produce a lot and you have a lot of waste products. In other organisms like plants and fungi, like for example the yeast Saccharomyces cerevisiae, that maybe some of you have heard of, because it is the one that is used to produce beer, for example. It is a slightly different pathway, but the same goals are there. You need to replenish NAD+, and you need to remove pyruvate in order to go on with the process. So basically the same thing will happen. Two ATP molecules will be produced from glycolysis, but pyruvate will get will in fact get the H plus from the NADH in order to reform the NAD plus by combining into ethanol after one CO2 molecule has been lost. So this is the ethanol fermentation, or the production of alcohol. The good thing with ethanol is that ethanol is able to cross the membrane, the cell membrane, so it will be easily removed from the cell, while with the lactic acid that we saw in animals and bacteria, it is slightly different because it doesn't cross the membrane very easily, so it will build up in the cell and eventually give rise to acidosis, which is the increase of acidity in the cell, or even an osmotic effect that will drive water into the cell. So it's not very good to have lactic acid accumulations, but ethanol in this case is easily removed from the cell. Now, a little metabolic integration. So, how are all the fuel molecules used by cells? How do cells regulate their use, basically? First of all, glucose will not necessarily be used for energy production all the time. Basically, if you have high amounts of ATP in the cell, there is no need to spend energy and to spend time to do more ATP molecules if you already have a lot. So in this case, it will be stored, glucose will be rather stored in larger molecules like glycogen in animals or starch in plants. And it's oligosaccharides that will store glucose molecules but make them readily available if you need to obtain energy when you're deprived of glucose. In contrast, when the cellular ATP is low, then glucose will not be stored and will, in contrast, enter glycolysis. Other molecules than glucose can be used for energy. First of all, we can use disaccharides in which glucose is found, so it just needs to separate the glucose molecule from the other molecule, and you will be able to enter in the glycolytic pathway. And it is also possible for other monosaccharides to enter the glycolytic pathway at a different step of glycolysis, like for example fructose, when it is phosphorylated, can enter directly in the second stage of glycolysis. Fatty acids can also be used in order to produce energy. The overall process is called beta oxidation. If you notice here, at the end of the fatty acid molecule is added a coenzyme A, exactly like what we had in the citric acid cycle. So basically the coenzyme A will bind with the end of the fatty acid. It will, in fact, a reaction will be catalyzed so that you remove this into the acetyl-CoA. In fact, the acetyl-CoA will separate itself from the fatty acid tail, bringing with it one of the carbons of the fatty acid. So each carbon will give rise to one acetyl-CoA that can enter in the citric acid cycle and produce the many NADH and ATP molecules. Meanwhile, just the removal of the coenzyme A from the fatty acid will itself produce two electron carriers that will also enter in the electron transport chain. So it's quite a lot of energy that can be gained from a single fatty acid, because each carbon will give rise to that. So fatty acids, as we just said, can enter directly in the TCA cycle or a citric acid cycle by the means of acetyl-CoA. It is also the case for amino acids like alanine, phenylalanine, isoleucine, or tryptophan that can be converted into acetyl-CoA and enter in the citric acid cycle. Glutamate and isoleucine, methionine can enter at various steps of the citric acid cycle, as well as, for example, tyrosine and aspartate that can all enter at different stages of the citric acid cycle. So you see here that all the amino acids forming proteins can also enter the citric acid cycle and give rise to energy. How is this all regulated? So we will play here with enzymes that are called allosteric. So if you remember the chapter on enzymes, we said that some substances could change the shape of enzymes and make them more or less active. It is the case here with allosteric activators and allosteric inhibitors. Basically, what happens is that when you have an accumulation of substrates like NAD+, ADP, and PI, so no products produced, then the accumulation of substrates will turn on all the enzymes of the chain and stimulate the glycolytic pathway. so that you will remove the NAD plus and the ADP, and in contrast form ATP and NADH. In the same order of idea, when you have an accumulation of ATP and NADH, it will turn off all the system because they will act as allosteric inhibitors that will switch all these enzymes off. If you look at one of these enzymes, like the phosphofructokinase, which is one of the first enzymes in the glycolytic pathway, you see that ADP or AMP, which are the substrates of ATP formation, will, when they are present in high concentration, activate the phosphofructokinase and turn on all the pathway by the production of fructose 1,6 bisphosphate, which is the second step of glycolysis. In contrast, when you have a lot of ATP, what happens is that the same enzyme will start to stop its activity and completely end the glycolytic pathway. Now, a couple of integration questions for you. Here are several questions that I would like you to think about. Intestinal cells need energy to maintain their sodium gradients, which are pumps that consume ATP, but they will never consume glucose. Can you explain me why? Also, red blood cells, they have no mitochondria. Why is it so? Lactic acid increases in muscle cells after a strenuous exercise. Why, and why is it painful? Adrenaline stimulates the release of glucose from glycogen in liver cells. Why, and how does it work? So, I hope that you will think of these questions and have answers for me tomorrow.